Cell communication is incredibly important to single cell and especially multicell organisms being able to communicate between different parts of the organism and maintain homeostasis. There are different ways that cells can communicate with themselves and each other. The first is called autocrine, auto meaning self. So this is when chemical signaling molecules are made by a single cell, which releases those, and then those chemicals have an effect on the same cell itself. Whereas in the next type, we have signaling across gap junctions. If you remember to our conversation about gap junctions, these are passageways between two fused membranes of two different cells in which one chemical can pass directly from a single cell to another cell. So we have the signaling cell and we have a target cell. In the next two cases, the signaling molecules need to be released from the originating cell, and they have to travel to their target cell and be recognized by that target cell. So in both paracrine and endocrine types, the signaling cell is going to create a signal molecule which will be released and has to travel to its target cell and be recognized. In the case of paracrine, the target cell is a nearby cell, so the molecules don't have to travel very far, very close by. Whereas in endocrine, the signaling molecule has to travel a distance and has to travel in the bloodstream to its target cell. So here's an example of paracrine signaling where you have a originating chemical messenger produced in one cell that's going to be released from that cell, have to travel to a nearby cell, not in the bloodstream, but travel to a nearby cell, bind to a receptor, and then trigger a response in its target cell. So here we have two nervous cells, a presynaptic cell, so before the space, this is the space, and a postsynaptic cell, this is the second cell after the space between the two. The presynaptic cell is going to release a neurotransmitter through vesicular transport. That neurotransmitter is going to go across the synapse or the space, bind to a receptor, and trigger a response in the postsynaptic cell. Now, as I mentioned, those second two types, both paracrine and endocrine, require that the chemical molecule bind to a receptor. So those receptors can either be inside of the target cell, or they might actually be on the outside, on the plasma membrane. In this first case, we'll look at internal receptors. So here we have a signal molecule coming to its target cell, and it's actually going to go through the target cell membrane, find its receptor in the cytoplasm, bind to it, and then usually that will allow it to go into the nucleus and have an effect on the transcription of the DNA. So we're usually talking here about hydrophobic molecules or lipid-based molecules that are able to go right across the plasma membrane, find the receptor inside of the cytoplasm, bind, then travel across the nuclear membrane, which is also lipid-based, and have an effect on the DNA to regulate expression of certain genes. The other kind of receptors are found on the surface of cells. So here we have for molecules which are hydrophilic, which are water-loving and not lipid-loving, these are molecules that cannot easily get across the plasma membrane. We need to have a receptor on the outside surface of the membrane for it to be able to go across and for it to be able to be recognized and send a message across the cellular membrane. So we're going to look at three types of cell surface receptors. One that's called ion channel linked receptor. So the receptor is linked to a channel which will let ions go across. A G protein linked receptor and an enzyme linked receptor. So these are 3 types of cell surface receptors. So in our first one, we have ion channel linked receptors. We talked about the fact that there are integral proteins, they go across the membrane, that when they are triggered will open up and let ions go through, because ions are very hydrophilic and they can't go across the membrane itself. They need to go through a channel. So in this case, the signal molecule will bind to a receptor site, and this will allow for the channel to open up and let ions go through. The second type is called a G protein linked receptor. G protein linked. So here is a G protein coupled receptor. So here is the whole receptor, and we have the G protein, which is heterotrimeric, which means it's got 3 parts to it. And associated with it is a GDP molecule. Kind of sounds like ATP, but it's similar, but it's a guanine-based molecule. So when the signal molecule binds to its receptor, so here we see the signal molecule is binding to its receptor, then the alpha subunit of the G protein is going to swap out the GDP for the GTP. Then that's going to trigger the dissociation of the gamma and the beta units from the alpha unit. That's going to trigger a response. Then the GDP is hydrolyzed, is broken apart to release a phosphate and to go back to the GDP, so that brings the receptor back into its original conformation. So it's only when the signal molecule binds do you get this cascade of events that results in a response inside the cell, because the signal molecule can't go inside the cell. And then the third type is called enzyme-linked receptor. So here we literally have a receptor and a tyrosine, a kinase receptor. That's this particular kind of receptor. And when the signal molecules bind to 2 of these tyrosine kinase receptor molecules in the membrane, then they become phosphorylated, they dimerize, they become phosphorylated, and that's going to result in a cellular response. So the receptor, tyrosine kinase, the enzyme-linked receptor, has a single transmembrane region. The binding of the signal molecule on the outside receptor, because remember these signal molecules can't get across, causes the receptor to come together to dimerize. Di means 2. To come together and make 2. See how they're farther apart up here? We're going to have them join together. The tyrosine residues are then phosphorylated. We're going to put phosphates onto the tyrosine residues, the tyrosine molecules, and that's going to cause the cellular response. And then there will be a phosphatase. That's an enzyme that will take these phosphates off. Once those come off, the 2 molecules will come apart. The signal molecule will drift away or be degenerated. So we've started talking about these signal molecules. What exactly are they? Well, they come in different types. We can have hydrophobic ligands. These are the signaling molecules. For example, steroids. So lipid-based, like we have estradiol, testosterone, cholesterol, those are hydrophobic. Those can go right through the plasma membrane and find their receptors inside. Then we have water-soluble ligands, for example, things like peptides and proteins. Here we have an amino acid, a peptide is a small protein, and then we have a larger accumulation of amino acids being proteins, and those are going to require a extracellular or a receptor on the outside. Additionally, we can even have ligands or signaling molecules which are made up of gases like nitric oxide. So now let's look at propagation or movement of a signal. So signal transduction is the case when that particular signal molecule or ligand attaches to its receptor, and that signal gets transferred through the cell membrane and into the cytoplasm in order to continue the signal. So here the signal can't get across the cell membrane because it's lipid, the cell membrane is lipid, so it needs a way to transfer that message across. Then the signaling pathway, this is a chain of events, kind of like dominoes. So when that signal molecule binds, it's going to set off a series of events, which often include activating things called second messengers, enzymes, or activated proteins that follow. That happens after the signal molecule binds to their receptors and are going to continue the effect inside the cell. Now, signal integration, sometimes you can have two or more signals on different cell surface receptors that actually interact with one another to cause the same response in a cell. So here we have an example of a signaling pathway, and we're going to talk about epidermal growth factor receptor. So you sometimes see just EGF, that's epidermal growth factor, causes the growth of epidermal tissues. And here, if it's got the R on it, we're talking about the receptor. So here is the epidermal growth factor receptor. It's one of those tyrosine kinase receptors. It's an enzyme type receptor. When epidermal growth factor binds to it, remember, we're going to activate these receptors, dimerize, cause phosphorylation. Two proteins are associated with the receptor called GRB2 and SOS. These two are going to activate what is called a RAS, and a small G protein. So this is going to set off this cascade of activation in which this GTP is going to get converted to GDP. And then a protein kinase is going to come in, this RAF. Once this RAS GTP is activated, then that is going to phosphorylate MEK. MEK, here it is phosphorylated, it's got the P stuck on it. That's going to phosphorylate the next kinase in line, the ERK. That's going to stimulate transition. It's going to enter the nucleus, and it's going to trigger these kinds of responses: cell proliferation, cell migration, adhesion, angiogenesis, which is the growth of new blood vessels. These are the kinds of things that epidermal growth factor stimulates. So the epidermal growth factor is the EGF. The receptor has the R on it, and this is a tyrosine kinase that is involved in regulating growth, wound healing, and tissue repair. When the EGF binds to its receptor, remember it's a tyrosine kinase receptor, and I'd like you to remember that it's one of those enzyme kind of receptors, it's going to set off this cascade of events, which is eventually going to end up in causing the cell to grow and get ready to divide. If this EGFR, this epidermal growth factor receptor, is activated at the wrong times, then you can get uncontrolled cell growth. Another name for that is cancer. So don't be so concerned about memorizing like all these different steps. Just know that the binding of this signal to its receptor sets off a cascade of phosphorylation events that eventually results in the cell making changes. Here is a YouTube video that you can watch, a short video that you can watch that will demonstrate this process of cell signaling. So now let's look at these methods of intracellular signaling. We talked about the fact that many of these steps involved phosphorylation. So remember that a phosphate group is a phosphate, with some oxygens bound to it. It's very reactive, and it gets added to nucleotides like GMP. So if we add one phosphate to a guanosine monophosphate, then we get a GDP. If we add another, we get a GTP. So when the phosphate group is added to residues of amino acids like serine, threonine, and tyrosine, eventually we're going to use that to add those on to those amino acids. And we're going to need a kinase to do that. So a kinase is an enzyme that catalyzes or stimulates the transfer of a phosphate group. Now, additionally, intracellularly, we may need these things called second messengers. So second messengers are involved in removing two phosphate groups from ATP to form a new molecule, which is called cyclic, that's what that C stands for, AMP. So adenosine monophosphate, we call it cyclic because when we have our ATP molecule with three phosphates, if we use this adenylate cyclase, this enzyme, to take off two phosphates, we end up with the last phosphate making a circle of bonds. So it's called cyclic AMP. Cyclic AMP is a second messenger. Why do you suppose it's called the second messenger? So let's see if you can figure out why it's called the second messenger, and we'll talk about that in class. So here we have some other second messengers. So phosphoinositol phosphate gets converted by phospholipase C into inositol phosphate and diacylglycerol, and both of these are two additional second messengers in addition to cyclic AMP. So we have three kinds of second messengers. We have cyclic AMP, diacylglycerol, and inositol phosphate. So what are some things that are responses to this signal molecule? The one thing that can happen is that gene expression or the reading of genes can get changed. So let's take a look at when ERK, which is a MAP kinase, so it's an enzyme, this that activates translation. So remember, translation is the making of proteins when it is phosphorylated. So here we have a number of different items associated with the messenger RNA, and when this gets phosphorylated, we're going to activate the reading of the messenger RNA so that we can make proteins. So the ERK phosphorylates the MNK1, which in turn phosphorylates the next step. So this one gets phosphorylated. First the yellow one gets phosphorylated, then the EIF4E gets phosphorylated. And when that becomes phosphorylated, the messenger RNA unfolds. See how it's got this pin, this hairpin in it, and it's going to unfold. And then allowing protein synthesis to actually start beginning. Some other responses to cell signaling. We can have an increase in cellular metabolism. So when adrenaline, which is a hormone, activates beta-adrenergic receptors on the outside of membranes, this will cause an increase in cyclic AMP. That's one of our second messengers. That's going to activate pKa, which is protein kinase A. pKa phosphorylates two enzymes, which lead to a supply of glucose and an increase in metabolism. Cell growth is a response to cell signaling. Growth factors bind to tyrosine kinases, like we saw with epidermal growth factor. These can initiate a pathway which activates the MAP kinase pathway, and that includes G protein called RAS. The MAP kinase stimulates protein expression that usually leads to cell division. So when we need cells to grow and get ready for division, then we're going to want to stimulate with growth factors through tyrosine kinase pathways. The next thing is an interesting topic. It's called apoptosis, or cell death. Now you might think that cell death is not a good thing, but in fact when it's programmed cell death, it's absolutely essential, especially to developing organisms. So here what you're looking at, this is the foot pad of a mouse embryo. And you can see that we obviously need to have more room in between these toes. And if you look at your own toes, you should have spaces between those toes. Well, when the organism starts out developing, it's just one big blob. So we need to have a component which will actually cause the cells in between the individual toes to be eliminated. This process is called apoptosis. So there's cell signals that will actually cause certain cells to die. And these are very important because we need to have certain cells being stopped during developmental progress. So we've mostly talked about communication between cells of multicellular organisms. Let's take just a few minutes and talk about signaling between single-celled organisms. And here we have some individual yeasts. So each one of these is its own organism. And yeast cells will communicate between each other by releasing a signal molecule that's called a mating factor. The mating factor binds to the nearby cells on surface receptors, and they will stop their normal growth cycles and initiate a cell signaling cascade that includes protein kinases and GTP binding proteins that are similar to G proteins. So they can communicate to one another when they need to make more yeast molecules. And here we see cell signaling in bacteria, which are also single-celled organisms. So the population density is an important factor that utilizes signaling. So it uses molecules that are called autoinducers, which are represented by these little red dots here. So when the cell density is very low, then these autoinducers, when they get released, they kind of just drift away from the cells. But when the cell density is high, there are more and more of these autoinducers. They're going to bind to receptors that they find inside the cells. They're going to trigger genes which are going to which are responsible for making more of the autoinducers, and these are going to lead to more cell divisions. So this is going to lead to higher and higher numbers of cells in what's called a positive feedback loop. So you can see that communication between cells, whether you're talking about a single-celled organism or a multi-celled organism, is going to be very crucial to the survival of the organism.